The sir with the black blazer, thank you.
Hi, I'm Neil Osterweil with MD Edge for Dr. Swanton. When do you intervene? In other words, how do you identify when it's time to intervene in the tumor formation process, and what's the biomarker?
So I think the key thing to say is that we don't know for certain whether this is a hit-and-run mechanism or not. We think it is. In other words, the release of IL-1 beta has a transient effect on the epithelium that creates this progenitor pool of cells. And if the cell harbors a mutation, an oncogenic mutation that happens to be present in normal tissue, and we estimate in a healthy adult, we may see about 1 in 600,000 cells in the lung harbor an oncogenic mutation, then a tumor could form. Now, the difficulty here is estimating or predicting who has a mutation and who doesn't, because these mutations are ubiquitous. They're a natural result of aging. We show in our study that the number of mutations that are detectable increases with age. But what we can't do with 1 trillion cells in the lung is sequence every cell and tell you which cell is vulnerable and which cell isn't. So that remains an intractable problem. So I don't think, you ask about a biomarker, that we're going to get a biomarker that tells us what's the at-risk cell. So we don't have a biomarker in truth at the moment. We know that IL-1 beta is released in response to pollution due to macrophage infiltration deep in the lungs, as well as from the epithelial cells we see release of interleukin-1 beta. We don't know if that could be a biomarker. I doubt it, because I think it will be very low levels detectable that won't be readily detectable. So the truth of the matter is that we would be looking probably at risk templated from the concentrations of PM2.5s in the atmosphere. But it's important to note that the relative risk here is probably an order of magnitude less than tobacco smoke. So we're probably talking risk levels somewhere between a 20% increased risk and perhaps 2.5-fold increased risk, depending on the concentration of PM2.5s in the atmosphere. Smoking, to give you an idea, would be 20 to 30-fold increased relative risk. The difference, of course, is that 5 times more people are exposed to what the WHO would classify as unhealthy or unsafe levels of pollution levels in the atmosphere globally, compared to the number of smokers. And of course, we have no choice of the air, over the air we breathe.
So we go to the online question from Stephanie Benz.
Stephanie Benz from L'Express in France. Another question for Mr. Swanton. Do you know how much time is needed and quantity of exposure is needed to have the start of a tumor?
Sorry, how much time?
The time delay between exposure to...
Yes, it's an excellent question. We're working with some collaborators in Canada on this question, Stephen Lamb and Renelle Myers, who published some work, interestingly, at World Lung Conference last month. And they showed they've got some tentative evidence that looks quite compelling that when they look at lead times of exposure of pollution, they're suggesting from their data that the minimum lead time where they can actually detect a signal is about 3 years of PM2.5 exposure. That's unpublished data, as is our work at the moment. But they suggest 3 years of PM2.5 exposure. That's work in progress. We've got more work to be done there, as well as we need more epidemiological cohorts to be able to demonstrate that, formally improve it. But it looks like it's possible that 3 years may be sufficient of exposure.
Thank you very much. Question here, second.
Hello there, it's Liam Davenport from Medscape. So I wanted to ask Dr. Kasper about you've obviously had active treatment versus placebo, but what was happening to the patients whilst they were on that treatment? What was the background? And you said that some people had had prior treatment and were recurrent. So what was the prior treatment?
So, I mean, the primary endpoint was PFS, and of course, and the patients in the placebo arm had the chance to receive the drug. So there was this open label neurogazettat extension in the case of disease. It is not a formal crossover, but it's more or less the same, because the patients had the chance, if they were progressive, they could they could be treated with a real drug.
But just to clarify, what was their management whilst they're on placebo? Were they just being observed? Were they just being followed up? That's what I'm trying to say.
You mean yeah. I mean, what we usually recommend in desmoid tumors, if there is no progression, we recommend active surveillance. And and that was actually the main point in the inclusion criteria. We really required RECIST progressive disease, so they had to have at least 20% progression of the tumor size to really sort out those patients that are in need for treatment, because there are quite a number of patients with more or less stable disease, or we also know about spontaneous regressions in this disease, but these were, of course, not the patients we wanted to have in that trial. So that's the reason why these inclusion criteria were quite strict, and to really sort out that we have the right patients and the patients that are in need for treatment.
Thank you very much. Any question from the audience? The lady here, third row, in blue.
Thank you. Annette Brindle from BioWorld. Another question for Dr. Casper. Can you give us a little bit of background about that drug and its first life in Alzheimer's, where I think it failed due to toxicity? And in general, these Notch inhibitors are pretty toxic. Can you talk about how that works with the desmoid tumors?
It actually at the end worked quite well with the desmoid tumors. So about the background is what we know is gamma secretase inhibitors inhibit Notch, but the full mechanism is not yet understood. But actually, it is the first drug in this disease where we know that there is a rationale behind that. I mean, we use tyrosine kinase inhibitors in desmoid tumors, but we actually didn't know how that works. They work in a way, but we don't know actually why. And the problem is with these other drugs, they are of course much more toxic than this drug. So it's more or less quite well tolerated overall. Of course, we have these side effects such as diarrhea, rash, and stomatitis. These are more or less the most important ones, but it's overall well tolerated. Patients could have the drug over a long period of time, and that was well manageable.
Thank you very much. Question, the lady here, please.
Hello, Virginie Bagouet from APM News in Paris. I have two questions to Dr. Swanton. Can you tell us if the protective effect of the canakinumab was observed on cell culture or directly on mouse? And can you tell us if the incidence of the non-smoker lung cancer is increasing, or is it how much?
Yeah. So I'll answer your last question first. It turns out we're not very good at collecting globally smoking data from patients with lung cancer. So it's actually quite hard to establish whether the absolute number of never smoking lung cancer cases is increasing. The proportion is increasing relative to smoking lung cancer because fewer people are smoking. But the jury is out, and it's quite controversial at the moment whether the absolute number incidence of never smoking lung cancer cases is increasing. Your first question, can you just repeat that, because I couldn't hear it very well? Canakinumab?
There's you talked about canakinumab,
yeah.
The antibody,
yeah.
And I didn't understand whether you actually observed a protective effect on cell cultures or in mouse.
No. Protective effect in mouse, in our mouse model of pollution-induced lung cancer.
Sorry, and I have questions to Dr. Hannon. I thought that the TIL therapy has already been studied in the early 90s. So what happened? Why did it take so long to arrive in phase 3? Is it really different from the one used in the 90s? And also, I would like to know how long does it take, how long the protocol takes, how long the people stay in hospital?
Thank you for these questions. Yes, TIL was developed by Steve Rosenberg at the NCI in the 1890s of the past century. I think the real trial that they did, where more or less the same protocol was used as we are currently using, was published in 2002 by Mark Dudley of his group in Science at the time. And then after those years, there was a lot of small trials ongoing because it's still complicated to grow these cells. So it probably took a long time, and then of course the checkpoint inhibitors came to the market, and that flooded everything, right? There was no interest in TIL at that time. So when we started the study in 2014, only ipilimumab was approved, and then later on the other drugs came also approved. We use exactly the same protocol that Rosenberg used in his publication of 2002. There was no change in that. So it's an old protocol, but it's very, very efficacious, as you have seen. So what is the time for the patients? The patient need to undergo surgery. And then in the time that they recover from that, because we need a metastatic lesion to grow the TIL, and if that can be a subcutaneous nodule, it's easy, of course, but if it's, let's say, a lung nodule that you want to take out, of course, patients have to undergo also sometimes some bigger surgery to do that. So they have the possibility to recover in the time that we grow the cells, which usually takes about 5 to 6 weeks. So the total period of treatment consists of about 8 weeks, more or less.
Thank you very much. Questions from the audience? No? So then I would have actually done a question into this context.
There's one more.
Someone?
There are two more questions over there, yeah.
I didn't see you. Please raise your hand so that I can see it well. Thank you very much. The lady here in blue. Third row there, and then we go back afterwards to you.
Thank you. Another question for Dr. Haanen. In the press release that we got, too, you said that you are trying to or will try to develop these without the without involvement of the biopharma industry. Is that correct?
Yes.
Can you talk about how that would work? And I know that colleagues of yours are doing similar things with CAR T cells. I'd be curious to hear about that whole effort.
So in this trial, no pharma was involved. These were point-of-care productions performed at NKI in Amsterdam and also at the Herlev Hospital in Copenhagen, Denmark. So it shows that it's very well possible to do this without any pharma involvement. And one of the reasons why there was no pharma involvement was there was no interest from pharma at that time that we started this study. That has changed. There are now a couple of companies that are making TIL and are treating patients with that, not only melanoma patients, but also non-small cell lung cancer, cervical cancer patients. So that is also now received interest from pharma. And I think the nice thing about the possibility to do this as a point of care is also that the price for the product will be different, right? So we can do it, as a not-for-profit organization at cost price, maybe a little bit more than that, also to make it sustainable. But of course, pharma will not do that because they have other obligations to their shareholders.
Thank you very much. There is the question from the lady here.
It's related to the other question. When do you think it could be available?
I think it's an important question to address. So within at least in the Netherlands, we have the study was supported by the Ministry of Health. So they paid for the cost of this study, and we made an agreement that if the study would be positive, then as of January 2023, it would just be standard of care in our country. Now, of course, in order to increase let's say, the availability of this treatment, one would have to go through EMA registration or FDA registration, and that is something that we are currently considering. But of course, that needs some more time. We are discussing with EMA to go this direction. If we would get EMA approval, of course, then the next question is, how do you roll it out over other countries, right? And then again, you get into the possibility that you either involve pharma to do this, or you train other centers, harmonize the protocol, and continue with the point-of-care productions.
Question here.
For Dr. Casper, I just wanted to return to you. I wanted to find out whether you're continuing follow-up, because I noticed that the median overall progression-free survival hasn't been met yet. It's not been achieved. And I wanted to ask about the degree of crossover as well.
Sorry?
I wanted to ask about the degree of crossover between the two arms.
Yeah. I mean, of course, we will follow up the patients. There are now, I think 84 patients are still in the open label nirogacestat extension arm. So after, and for the crossover that was, or for the open label, no, it was not a real crossover. I mean, again, it was all patients had after the placebo patients, when they had resist progressive disease, they could have the right drug, or the verum, and of course the other patients that were already in the nirogacestat arm could of course continue with nirogacestat in the open label extension phase.
We cannot hear you. You don't have the mic. Sorry. And then afterwards, we will go to our question online, please.
Sorry, so how long do you envisage people taking the drug for?
I mean, usually you take the drug until, or as long as the patients benefit from the drug. That means as long as there is no progression, or if there is symptom improvement and so on. So we have, from the earlier trial, from the small phase 2 trial, we have patients that are already on drug for years now. But of course, that is a very important question at the end, which is of course not answered by that study. How long should we treat our patients? And for that, we definitely need another trial. A discontinuation trial or something like that would be interesting for that, of course, because that's something we actually cannot answer with that trial.
Thank you very much, Bernd. There is a question from online. Alain Perez, no, sorry. She's not anymore with the hand up. Any hands up? Yes, please, first row.
Thank you. Hans Andersen from Health Talk Norway. John Hannan, you compare towards ipilimumab, but for at least people with quite a good health, they're treated with EP plus nivolumab. Can you say a little bit about your results towards the combination therapy?
Yes, that's an important point. Over the past years, right? This is really a treatment paradigm that is changing over time, right? And the study ran for 7.5 years, so things have changed. So originally, ipilimumab was the only treatment that was available for the patient who had failed anti-PD-1, but more recently there are data emerging that also ipilimumab plus nivolumab can still overcome the refractory patients after anti-PD-1. There are retrospective data on that, and there is one prospective study that was presented at ACR this year, the SWOG 1616 study, and that was a relatively small study comparing ipilimumab to ipilimumab plus nivolumab. And there the objective response rate was about 28% for the ipi-nivo arm and about 9% for the patients treated with ipi alone. Now, if you compare that to our trial, you would say, well, you know, TIL is much better, but the patient populations were not completely the same, right? That was let's say, a patient population that had probably more advanced disease, higher LDH, so you cannot compare them directly. But I still think that even if you correct for all of that, TIL is still a very interesting treatment option, even in comparison to ipilimumab plus nivolumab.
Thank you very much. any further question? Before there was Stephanie Benz with an open question online. She's not anymore. Yeah, she has the hand up again. Please.
Yes, another question for Mr. Swanton. You said that not everybody with mutation and air pollution exposure would get cancer, and that it was an open question to understand why. But in your hypothesis, do you have what is going on with the immune system, for example, of the person who don't get cancer?
So sorry, I'm struggling to hear the question, but I think what you said is, what is the relevance of the immune system in the induction of cancer? Is that right?
Is it your hypothesis to explain why people with mutation and air exposure, some people won't get cancer?
I didn't quite understand the question.
Really
if you can repeat it in a short time, because we have 4 minutes left. If you can repeat the question. Or eventually
I will send an email. Sorry about that.
Yeah, okay. Send me an email and I'll respond by email. Good idea. Yeah.
Otherwise, you can write it in the chat and I will read it. Stephanie, if you have not the question is I couldn't understand. I'm sorry. The question. No, okay. So we have left 2 minutes left for the last question. So maybe very briefly, we have 3 minutes left. Charles, one question. What is the next step for prevention now?
Well, I mean, I think there are over 200 environmental carcinogens. And if you go by the Alan Bell main data, the majority of environmental carcinogens that they studied did not induce DNA mutations. So the question is, how are they inducing cancer? And I'm pretty certain it'll turn out they're working through this promotion model that Berenblum first proposed in 1947. So this presents an opportunity for us to understand. I mean, I don't think they'll all work through IL-1 beta, I'm sure they won't, but there'll be different inflammatory networks in different tissues. And the challenge now is to tease those apart and work out which ones we could potentially target to limit risks of cancer in exposed populations if we can't lower the environmental carcinogen in any other way. And then the second thing is to understand that with tobacco and UV light, for instance, are they also acting in a promoting fashion? Of course, they're causing mutations, but that doesn't exclude the possibility they're causing cancer as well through promotion. And obviously that has public health implications, because we've always assumed, for instance, that tobacco is just causing mutations and nothing else. But if it's causing pollution-induced inflammation in the lung, then that starts to question, in my mind, the safety of vaping, for example, which could also be having a similar inflammatory effect in the lung. And I think we need further studies on vaping to see if perhaps vaping could act as a promoter too.
Thank you very much. It has been exciting news, I have to say, and I hope you have been, as well as myself, I'm very much excited by hearing all of these information. Thank you very much for having been here. I wish you a wonderful ESMO 2022.